not leave the tubes unless they are forced. For example, during the mating, even a male enters into the female tube. So after the mating, females lay the eggs into the root pouch, and the root pouch is made of plates, called the osteocytes, which grow up from the base of the theropods. So inside of the root pouch develops the eggs, and from eggs develop into a juvenile stage, called the manuka. So I like here emphasize that canines do not have a planktonic larvae at all. They do not have a larvae at all. Just from eggs develop juvenile stage called manuka. So manuka looks exactly as adult stage, but it only lacks the last pair of theropods. So when manuka leaves the root pouch, so it stays with the female in one tube, and once it is ready to leave the tube, drill the pores in the wall of the tube of the female, get out of the tube and build its own tube next right to the female. So this lifestyle, non-planktonic stage and immobile manuka, so gives us idea that canines have a really limited tertiary potential. And for this reason we think that they are ideal indicator of environmental condition, just because canines stay where they are unless they are formed by environmental condition like a physical, chemical, biological condition. So I said that canines do not move much, but we have some of the exception. First I would like to explain that we have at least 3 different types of different males. First male is called a terminal male. We call it a terminal just because it doesn't fit, so it's had a reduced mouth parts. This may usually have better developed claw and the pleopods. Sometimes the claw are really big, but anyway, this male usually share a species-specific character with the females. For example, here in Neocanines capsicata, there is a row of a city on a Chilipes carpus, and which is very unusual character for this genus. So we believe that this is the same species. And the same with Neocanines dimorphus. This is an Antarctic species, and the male and the female morphologically look different, but the secretion on the legs is really consistent, so they are paired. The second type of a male is what we are calling the juvenile male. It's probably sexually mature, but we call it a juvenile male because it looks like a juvenile female. So it has a little bit thicker antenna, a little bit better developed pleopod, but it's smaller than the female. And the third type of male is what we are calling a swimming male, and this is totally different from a female. So without molecular methods, we are not able to turn male and female. And last year, we have been for the first time able to identify 6 of the males we catch outside of Iceland in the deep sea. So these 6 males, we have been quite sure that they belong to a different species just because of the different shape of the chela, different shape of antenna. This has a spine on the tail, so it doesn't. So we were sure that we are dealing with 6 different taxa, but we didn't know what they are. And thanks to CO1 sequences, for the first time we have been we find out how it looks made of the family Typhlotanaide, Cryptococcidae and Agathotanaide, and also we identify male of Acanthophoridae, but we didn't find a female for that. So anyway, the most surprising for us was male of Agathotanaide, just because we knew that in this family juvenile male exists. So now we found a swimming male, which means that in this family we have 2 types of male. And we tried to explain that, and explanation is mating tactics, which is called female mimicry hypothesis. So juvenile male don't swim, so it's made with a female nearby. And so such a small male, which looks like a female, is less likely to be attacked by an adult male, but it's also less probably to be tolerated by the other females in the breeding area. And once they are molting, they begin the swimming male, so they can swim and mate with a distant female. So this looks like a strategy for a deep sea, because if we assume that the deep sea population is quite of living in a low abundance, so like inter-population connectivity might be a good strategy for surviving. And surely it's an evolutionary benefit. So this is what we know about the Clapeyron-Clipperton fracture zone. So there are of course the nerves download from the orbits, so there's not much record. And there is altogether 10 species described. I'm going to say a couple of words about the species and the problems which we have with identification of them and the classification of them. But before that, I would like to say what we could expect in that area. So first of all, we could expect two families of Apseudomorpha, Apseudidae and Spiratovidae, just because there are two families occur in a deep sea. This is how they look like. These are Apseudidae, usually very spiny, and Spiratovidae, less spiny but with hammer-shaped achilla and very long legs in males. And with Thanaidomorpha it's much more difficult, just because, let me explain. First three of this family are typical, they retain eyes and they are typical shallow water taxa, so we shouldn't expect them in a deep sea. Exception is Thanaido. They have, some of them have eyes, but it's a rule that if they live on a shell then retain eyes, if they live in a deep sea they are blind. So we might have this family as well. I would like to pay your attention that 8% of Thanaidomorphs they are genera Incertasedis, they are taxa, the species with family Incertasedis. And these three families, they are surely polyphyletic family. So it means that every, like 40%, every 4 species, in fact, we have a problem with identification of that and classification. So family is surely ensured. So that is the conclusion that if you would like to work with Thanaids on a level family, then surely that must be an artifact, because we are not sure of its system. Okay, so one of the species that have been found in Clapeyron-Clipperton fracture zone is Heliceticanalis spini-maxillipedis, very difficult name, which have been classified to family Coleteridae. But in fact is very, very similar to Paranarturella, the genus which we described, revised 2 or 3 years ago. And for now this genus is classified to Incertasedis. So we think that these two guys in fact are closely related and perhaps represent a new family. The other two taxa belongs to genus Agathocanalis. Agathocanalis is a quite a very distinct genus just because it doesn't have a second antenna, or actually has a bugs, very short second antenna. But these two species, this one has a very long, and this has none. So that might be closely related general to Agathocanalis, but not Agathocanalis itself. And another species has been classified to genus Stenothanalis, but the typical Stenothanalis, this is Stenothanalis from the North Atlantic. So the Stenothanalis has a carpal shield, very well developed, also has a very characteristic bent downward uropod. So this doesn't, so I think that they might represent a new genus called Stenothanalis. And finally, member of Typhlotanalis. The Typhlotanaidae family are defined by a presence here on the last three theropod, what we are calling the cleaning apparatus, which is a special apparatus which we believe to allow animals to be better kept inside the tube. But looking at the morphology of the general morphology, I would say this is not a Okay, so we have lots of questions in taxonomy and systematic, and what would help us would be a material which would be proper for molecular analysis. And I would like to say a couple of words about the project Ice Age, Icelandic and Marine Animals Genetic and Ecology, which was led by a secondary research institute in a cooperation with the University of Iceland. So during this expedition, the tanite material had been collected around the Iceland, and one of the gears was epibentic sledge. This epibentic sledge has two nets, and in the end of the nets there are what we are called a cod end. So that's what the animals which have been catching the cod end have been used for molecular analysis, so have been preserved in ethanol. All of the rest in the net have been preserved in formaldehyde and was much better for taxonomical research. So thanks of this project, we received 151 CO1 sequences, and I know that this result is maybe not very spectacular for most of you, but if we see that in a gene bank exists only 29 CO1 sequences of tanite, so this score is very, very good one. So we amended the logo of Ice Age. So what was the success? First of all, it was what we have been called a cool chain. So means that the material which have been kept in a cod end was immediately transferred to a cold room, so the room where the temperature was 4 degrees. The water was, for sieving, was pre-cooled to 4 degrees. The material was sieved on a sieve with a screen 0.3 millimeters. Then the material was preserved in the ethanol pre-cooled to minus 24 degrees. Then was stored in minus 20 degrees. After 24 hours, ethanol had this exchange. And then samples had been sorted on ice, so under the microscope, the dish packed with the ice, and also the tubes had been all the time fixed together with ice in a special bath, the tray, aluminum tray. So all of the time, samples have been kept cold. And this increased our success of receiving the sequences from 10% to 50-60%. And we received a tree. This is a tree made by CO1. As I said, I don't agree that neotanidomorphs might be fused with the tanidomorphs, because in my tree is absolutely in different place. So it's not that the system which we have of division tanites into 3 suborders is more or less correct, unless pseudomorphs are not monophyletic. But anyway, to study taxonomy, we need a signal from molecular biology, and to interpret molecular results, we need the taxonomy. So both of these things must be complemented. And finally, I would like to say about the IntKey, that's an interactive key for tanites. We did this key last year during our little workshop with my students and my colleague, Dr. Roger Bauer, and another colleague and 3 students. So we have been working for a couple of days, actually 1 week, at the field station, University Station. So this is our bag, it's on the bed as well. Okay, and we did interactive key using as a template Jim Lowry's keys for identification of amphipods. So I'm not sure how familiar you are with the interactive key, but this key allows you to start with the character you want. So you don't have to follow all the characters as usually in the key is. You can start with whatever character you want. And once you choose a character, so some of the families are immediately eliminated, the same some of the characters are immediately eliminated. So the advantage of the interactive key is that there is a visualization of each character. So for example, you don't have to look at the character and understand the character, you just to pick up the picture. So with tanites we didn't do that, but we have a list of characters, we have a list of the families and genera. So I am very happy to share this key with you during this workshop. Okay, and this is all what I would like to say. Thank you very much for your attention.
Thank you very much, Magdalena. Any questions? Yes.
Hi, thank you for your presentation. I was wondering, is the epibentic sledge the most ideal tool to collect tanites, or is box score way worse than epibentic sledge?
Epibentic sledge I think is better because it's given a much more material and in a better condition than box score. Box score is fantastic for ecological assessment, but just to get a material proper for taxonomy or molecular biology, the EBS is better.
Okay, thank you.
Any other question? Stefan?
Thank you. You mentioned that there is no larval stage and very little dispersal in some species. Wouldn't that mean that this is one group that could be easily impacted by mining, so it would be a good species to study?
Yeah, this is why we think that they are good bioindicators because they are living there, and in theory, their ability for dispersion should be really, really low. They do not swim, in theory. But how the species behave at a depth 6,000 meters is not idea.
Lenai?
But you also said that the males were able to swim, so the male can make the connectivity among the populations.
That's what we believe.
And do you know, are you aware of any study that has been looking at the connectivity among population of canaries?
But it would be very interesting. This is actually what we are interested in, how the species could be really distributed. What is the potential of the distribution? It's really interesting.
Yes.
You asked about ink file. This is new ink file because before probably already exists the old ink file for Delta program.
I am not aware of that.
Ink file for tanate identification using the Delta program. There's a new ink file.
This one?
Yeah.
Yes.
Yes. And also second question. What do you think? It is possible to create a special key book for the CCFZ tanate in this time?
I'm not sure. Probably no, probably not. But first, we don't know the taxa, we don't know what lives there, except this what I show. And well, but identification if the times might take a while, and to require the dissecting as well. So.
For example, if the contractor, maybe all contractor, maybe part of contractors, allow us, you, to work with tanate collection of their own, you can do this key book.
Well, the question is a time. The question is a time. Yeah, we can try. I don't say no.
Any other question? Comments? Thank you very much, Magda.
Thank you.
So we move to our next presentation. Charlotte Habermas.
Since we have one presentation less in the evening, I think we can make these two separate presentations right now.
Yes. No problem.
Okay, I thought I had part two. So I want to start first about using antibody, but ones exist. And actually these methods can be used in two ways. So we can have an integrated approach where we combine genes and morphology. Then morphological studies actually build on the results of the molecular ones. And this is also called reverse taxonomy. And for this I will highlight you some results of my own studies. And then secondly, we have what I call the blind taxonomic approach, where we actually treat species as molecular operational taxonomic units, and so their DNA stands alone. And then finally, I will show you that molecular tools cannot only be used for species identification, but also for questions which go beyond this and on general deep sea ecology, etc. So what are the challenges? First of all, as you know, there are incomplete species descriptions. Most of the species are new to science, as well as genera and sometimes families. And also molecular studies have already shown that in many deep sea taxa there are cryptic species, so species that are actually genetically different, but morphologically they look the same, they are the same. And so this makes it all more complex. And then there is another question, how do we evaluate interspecific variability? For example, when a species has been described from the Southern Ocean and we want to compare it to the Pacific and we find some small differences, are they small interspecific differences or are they interspecific differences? And also for sexual dimorphism, but I don't think for amphipods this is less the case than for other crustaceans. So this is just a summary to show you in general what as molecular studies have been conducted on deep sea taxa. And so just so that you can situate the work on amphipods, excluding the one I'm going to present now. So in the past, one always used allozyme studies to study gene flow, and this has been evolved until the use of fast evolving markers like microsatellites and also genetic fingerprinting with EFLPs, for example. And what we can also see here is that the modern use of next generation sequencing tools has not yet reached the deep ocean because, to my knowledge, there has not been no studies on this on deep sea taxa. And one method I will focus on is DNA barcoding. It is an initiative that has received a lot of attention during the last decade, but also a lot of controversy. And it's actually